idiopathic pulmonary fibrosis, or IPF. I mean, when you look at the diagnosis, it's just a terrifying prognosis. It's chronic, progressive, irreversible scarring of the lung tissue. The clock is absolutely running, and our current treatments are pretty limited.
They are.
And a big reason why is that we still don't fully grasp the underlying molecular events, the true pathogenesis that turns healthy lung into scar tissue.
And that lack of understanding is, well, it's compounded by a huge roadblock in research, which is translatability.
Right.
Our standard preclinical models, and we're talking mainly about the bleomycin-induced mouse model, they just don't perfectly mimic the human disease.
How so?
Well, the mice often heal, they actually resolve the injury, whereas the human lung just continues to spiral deeper into that irreversible fibrosis.
So if our traditional cell studies couldn't give us the context, we needed to know exactly where the damage was happening, down to the level of the cell's own neighborhood.
Oh.
And that's really what this deep dive is all about. We're unpacking just a revolutionary study that uses spatially resolved transcriptomics, SRT technology.
SRT is, well, it's arguably the greatest leap forward since single-cell sequencing.
Okay, so single cell tells us who is in the room, right? Which cells, what genes they're turning on.
Precisely, but to do that, you have to shred the tissue. You completely lose the architecture.
You lose the neighborhood.
You lose a neighborhood. SRT, using platforms like Visium, it performs that same RNA profiling, but on the intact tissue.
So you're taking a frozen slice of lung, you put it on this special slide that's covered in thousands of microscopic spots, and each spot basically takes a localized photo of gene activity.
A snapshot, yeah. Each spot captures the RNA from a small mix of cells right there, creating this high-resolution map of the entire disease.
And this study generated these maps for 4 human IPF lungs and compared them directly to that bleomycin mouse model.
At 2 really key time points, day 7, which is the acute injury phase, and day 21, the more established fibrosis.
Okay, let's unpack this. Our mission is clear. We need to use these maps to find the specific microenvironments, the fibrotic niches, and really zero in on the differences between the human disease and the mouse model. And we're starting with the star player in human IPF.
When researchers first started using single-cell in human IPF, they noticed some highly unusual cellular states, atypical fibroblasts, profibrotic macrophages, and most importantly,
this population of epithelial cells that are kind of stuck.
Stuck in an intermediate state, exactly.
And these are the famous, or maybe infamous, KRT $5 plus aberrant basaloid cells.
The ABBA cells, yes. And let's break that name down because it's so key to the whole problem. They're basaloid because they express KRT 7C17.
Which suggests they're trying to become a protective lung cell.
They're trying, but the really crucial part is the negative marker, KRT5.
Okay.
KRT55 is a marker for a fully functional, mature basal cell, the stable foundation of the airway. Lacking KRT555 means these cells they're arrested, they can't complete the repair process.
So they're like shapeshifters caught mid-shift. They have markers of epithelial cells, basal cells, even the mesenchymal cells that make scar tissue.
They are perpetually frustrated repairmen.
They're just pumping out the very material that causes the scarring. The ECM proteins, the source suggests they probably come from normal progenitor cells like alveolar type 2 cells.
AT2 cells or club cells, yes. Now, contrast that with the mouse. In the bleomycin model, you get a similar kind of cell, the KRT8 plus alveolar differentiation intermediate, or ADI cells.
But there's a critical difference.
A huge difference. The mouse ADI cells successfully complete the repair. The fibrosis actually resolves. The human ABBA cell, on the other hand, is the very definition of arrested regeneration.
So to handle all this spatial data, we're talking thousands of genes across thousands of spots. The researchers needed a really smart way to simplify it all.
They did. They turned to something called non-negative matrix factorization, or NMF.
NMF. Break that down for us.
NMF is a pretty clever data deconvolution technique. So instead of looking at every gene in every single spot, NMF finds patterns.
I like the analogy from the source material. It's like finding the core recipes for different tissue structures.
That's a great way to put it. It takes all that complex gene expression data and boils it down to, in this case, 30 distinct factors. And each factor is just a signature of genes that always appear together. They define a specific microenvironment.
So one factor might be the signature for a healthy bit of lung, another might be a blood vessel.
Exactly. And when they focused on the factors that were most active in the IPS samples, one stood out from the pack.
Factor 14. Farfourtime-5.
Farfourtine was the undisputed champion of pathology.
Why that one specifically?
Because it represented the true engine of that pathological remodeling. When they looked at the genes inside it, it was a heavy mix of keratins like KRT7 and KRT8, but also collagens.
Ah, so it's the perfect mix of epithelial stress and scar production.
It is. And crucially, they found that the activity of F4200 cells correlated perfectly with the density of those key pathogenic cells.
The ABBA cells.
The KRT5, KRT17 plus ABBA cells, the myofibroblasts, and the AS1 high fibroblasts. All the major scar makers.
That is just a huge finding. I mean, they're using a pure data reduction algorithm, and it independently validates the exact cell type that was found in previous studies.
And it maps its precise location in the lung. I mean, visual confirmation showed that the spots with high 514-1 activity lined up perfectly with fibroblastic foci.
The FF, the literal hotbeds of active scarring that pathologists can see under a microscope.
Right, and the ABBA cells were sitting right along the borders of these FF. It confirms they aren't just bystanders, they are spatially positioned at the leading edge of the scar, coordinating the whole chaotic process.
Okay, so let's just crystallize what we've learned so far. Human IPF is really centered around these ABBA cells, these arrested repair cells.
Right.
And their location, specifically around those fibroblastic foci, was definitively confirmed using that spatial map created by NMF factor 14.
And knowing the location, that's just the first step. If the ABBA cell is the contractor building the scar, we now need to know who's sending the blueprints.
What molecular signals are coordinating all this?
Exactly. What is happening right at that border? This is where the true power of these SRT maps really comes into play.
Which brings us right into the signaling. The researchers, they zoomed in on those F14ODR spots, the ones with the highest pathological activity, and broke them into 5 smaller subclusters, CO through C4.
Just to see the organization of that neighborhood. And cluster C0, that was the ABBA core, confirmed by its gene markers.
And the other clusters?
The neighboring clusters included specialized fibroblasts and myofibroblasts in C1 and C2, and then macrophages in C3.
It's just a beautifully organized picture of chaos, isn't it?
Yeah.
You have C1, the fibroblasts focused on laying down scar tissue within the fibroblastic foci.
While C0, the ABBA core, is enriched right around the edges, acting as that transitional zone.
The spatial mapping also gives some really powerful evidence for where these cells come from, their lineage.
It did. They saw the highest abundance of normal AT2 cells a little bit further away, but transitional AT2 cells peaked right next to the CO-ABBA core.
Reinforcing that idea that the normal repair path, AT2 to functional AT1, is getting derailed.
It's being derailed into this dysfunctional ABBA state. The cells just stop. They get stuck at that C0 ABBA stage.
And the genes confirm the malfunction. The genes for normal alveolar function, the ones we need to breathe, they were just silenced inside that C0 cluster.
And conversely, fibrosis genes like MMP11 and COL1A2 showed this clear gradient, super high expression at the ABBA core, and then it drops off the further you get away.
So let's talk about communication, the cell-to-cell signaling.
Using some pretty sophisticated network analysis, they found the usual suspects. Canonical profibrotic signaling was, dominant. TGF-β 1 and TGF-β 2 were predicted to be highly active.
Both in the ABBA core and the nearby fibroblasts, which, confirms the standard view. But what about the less obvious regulators?
Well, they also found TP533, which is linked to cellular senescence, and the TED144 family members.
Which are central to the hipposignaling pathway.
Right, which is a master regulator of organ size, regeneration, and also arrested development.
But the finding that really made me sit up was the role of apolipoprotein E. APOE.
Yeah, me too. I mean, this is a molecule we typically associate with lipid transport, maybe in the brain with Alzheimer's. But here, they found APOE ligands were highly expressed in that neighboring macrophage cluster, C3.
Suggesting a specific kind of profibrotic macrophage.
An M2-like phenotype, yeah. But here's the plot twist.
Okay.
The receptors for APOE, specifically GPC1, LRP1, and CFTC1, were highly expressed within that ABBA C0 core.
So you have a tight proximity-dependent signaling loop. The macrophages are sending out these lipid signals.
And the arrested epithelial cells are just avidly receiving them. It suggests a completely under-recognized role for lipid signaling in permanently locking these cells into that fibrotic state.
That's more than just a footnote. It's a potentially novel mechanism linking immune cells, lipid transport, and epithelial failure right in that niche. So to summarize this critical second section, the human fibrotic niche is driven by these traditional pathways like TGF-β and TED. But the spatial analysis uncovered a novel, proximity-dependent mechanism involving APOE.
Ligands from M2 macrophages, receptors on the OBDA cells, a signaling loop that just perpetuates the scarring.
So we've established this complex arrested repair process in humans. Now for the ultimate test. How does this compare to the mouse model, the one that's supposed to resolve?
And this is where we hit the translational gap.
Section 3. It starts with just the overall molecular signature.
And the results were, frankly, startling. The molecular overlap between the human IPF fibrotic regions and the mouse day 21 regions was extremely limited.
How limited?
Only 93 shared differentially expressed genes, but even more concerning, they found several key genes that showed contrasting directionality.
Wait, contrasting directionality? You mean a gene could be upregulated in human IPF, driving the disease, but it's downregulated in the mouse model?
Precisely that. Take COL17A1, which is involved in the ECM, or the inflammatory molecule CX3CL1. They are behaving in fundamentally different ways depending on whether the injury is chronic, like in humans, or resolving, like in the mouse.
So it's not just that the models are different. The pathways they engage can actually oppose each other.
That's right. And at the cellular level, the mouse model just confirmed successful repair. AT2 cell numbers drop at day 7, but they recover by day 21. It's actively regenerating a healthy lung.
So when they compared the equivalent NMF factors, the human ABA factor versus the mouse ADI factor, the divergence became impossible to ignore.
It really did. The human F14.4 correlates strongly with ABA cells and myofibroblasts, the pure scar makers.
Okay.
The mouse F14.4, on the other hand, correlates mainly with the ADI cells and normal AT2 cells. And critically, it also correlates with AT1 cells.
The fully functional, regenerated lung lining cells.
And that correlation with AT1 cells is completely missing in the human fibrotic niche.
So even though the mouse ADI and human ABA cells share some common starting points, their destinations are worlds apart. One is on a path to repair, the other is trapped in a cul-de-sac.
Functionally, the mouse ADI signature is driving inflammation and then repair. The human ABA signature is just defined by chronic failure and irreversible remodeling.
And the regulatory factors must reflect that fork in the road.
They do. They share TGF-β and TP500-301, so a common injury response. But the IPF niche is uniquely activating factors like the apolipoprotein receptor, sore leather. Meanwhile, the mouse niche activates regulators we associate with healing, like high PLO and IAPOL.
Which brings us back to that dramatic disconnect with APOE.
The most dramatic one. In the mouse ADI niche, APOE and its receptor, LDL, were predicted to be inhibited, downregulated.
The precise opposite of the activating role it seems to have in the human ABA niche.
The very signal that helps drive chronic fibrosis in humans is actively suppressed during successful resolution in mice.
That puts the final nail in the coffin for this standard mouse model, doesn't it? The cellular fate contrast is just the strongest piece of evidence.
It is. In the mouse, the path is continuous. AT2 to activated AT2 to ADI and then to functional AT1 cells. Full repair.
But in the human lung, the pathway diverges. AT2 goes to a transitional state and then it hits a crossroads. It can branch into either functional AT1 cells or into those persistent scar-driving ABA cells.
And the process is arrested right there. That ABA state is a persistent intermediate that just fails to complete the circuit.
And that divergence-arrested regeneration versus active repair, that is the entire thesis of this study. It's the core takeaway for you. The difference isn't just where the scarring happens, it's the stalled fate of the lung's own progenitor cells.
And this gives us an incredibly specific roadmap for new therapies. Simply blocking TGF-β might not be enough. We need interventions that can kick those ABA cells out of their arrested state.
Targeting the molecules that are unique to that human niche.
Exactly. Things involved in senescence like TP5-33 or failed differentiation like TED, and especially that weird APOE signaling loop, those are incredibly promising new targets.
The goal being to force those ABA cells back onto the regenerative path toward functional AT1 cells.
That's the idea.
Thinking about the future really begs the question, if the standard bleomycin mouse model heals too quickly, maybe the next step is to pivot to more challenging models.
Models that induce a more persistent senescent state, something like a repeat BLM installation model.
We have to match the chronicity of the human disease, not just the initial acute injury.
Absolutely.
So let me leave you with this final thought. Considering how fundamentally different the signaling pathways are for APOE activating in humans, inhibited in mice, what specific regulatory role do you think this lipid-related molecule actually plays in permanently locking human epithelial cells into that aberrant scarring state? It's a mechanism we urgently need to unlock.